It's a real pleasure for me to be here today. I'm coming from Boston, and I really want to thank everyone present here, and particularly all members of this Waldenstrom Macroglobulinemia Foundation of Canada, and for inviting me to this forum. It's a pleasure for me to present my work, and it's a pleasure for me to visit this beautiful place. I'm originally from Chile. If you find any particular accent, please pardon me. But so this is a work that we are doing in collaboration with Dr. Steve Trion at the Dana-Farber Cancer Institute. And the idea is to engineer a mouse model of lymphoplasmacytic lymphoma, Waldenstrom macroglobulinemia. And today, I have the difficult job of explaining to you why this work is important and why your investment has not been wasted and why we are going in the right direction with our research. The goal of this work is to engineer a clinically relevant mouse model of LPL Waldenstrom macroglobulinemia, which has been a long sought goal for the field. And I have to say clinically relevant because there are one or two models available. They were produced a couple of years ago of Waldenstrom. But these are not Waldenstrom. I think those models do not recapitulate the clinical pathological features that are essential to develop a bona fide model of Waldenstrom macroglobulinemia. In addition, I want to tell you that people have been using cell lines. And before, one of the presenters indicated it's very difficult to generate cell line for doing research in Waldenstrom macroglobulinemia. For this is the second reason why it's so important to generate this mouse model of Waldenstrom. The benefit of this model is the availability is that will allow us stringent in vivo validation of functional characterization of LPL Waldenstrom genetic lesion. All those lesions that you heard about that have been identified by genomic studies can be validated individually and understand the biology of each one if we have this model available. And the second most important point that is for you is that we can assess target therapies with these animals. The approach that we are going to use is the following. It's a very sophisticated approach, and I'm going to do my best trying to simplify this, and it has been a long and time-consuming process. The idea is to reproduce in mice in a systematic manner the most highly recurrent, co-occurring genetic alteration identified in LPL Waldenstrom patients. And I'm referring to two of the most important ones. The NYD88 that everybody has been talking today, and the chromosome 6 deletion. I'm a pathologist. I'm a clinician that unfortunately doesn't have contact with patients, but we provide a lot of support to our clinician. I'm one of those persons that when receive the biopsy from a patient, those bone marrow biopsies, we are the people that we stain those slides, we do section, and we do immunohistochemistry, and we are able to make the decision and say to the clinician, unfortunately, this is a patient with LPL, and this is the percentage of involvement in that bone marrow. And here I want to briefly show you how we do diagnosis in pathology, because this is important. This is exactly the same process that we are doing with our animal models to characterize and to say this animal is developing Waldenstrom or is not developing. And as you remember that Waldenstrom is a B-cell neoplasm, B-cell that proliferate, and those cells are characteristically CD20 positive, they are CD5 negative, they are CD10 negative, and they are IgM positive. In addition to those lymphocytes, it was said before that the patient with Waldenstrom do have lymphoplasmacytic cells and plasma cell that is very characteristic of this disease. And the plasma cells are less frequent than the lymphocyte and are characterized for being CD138 positive and IgM positive. And this is a particular case of a patient with Waldenstrom that has a high degree of involvement, in which you have about 50% involvement with lymphocyte and plasmacytoid cells and about 10% of plasma cells. When LPL is characterized by bone marrow involvement and IgM monoclonal gammopathy, is called Waldenstrom. And in those circumstances, we as a pathology, we diagnose those features because we see a spike of IgM, a spike in the serum. And also because we see in the bone marrow and in peripheral blood that the red blood cells aggregate and produce what is called rouleau formation. So all those features we use a pathology for making diagnosis in patients as well as in our mice. And you heard already about NYD88 is an adaptor protein that brings together signaling from the toll-like receptor and interleukin-6 receptor and channel down to activate a pathway that is very important for oncogenesis, that is NF-kappa B, and is characterized by translocation of a protein from the cytoplasm to the cell, from the cell to the nucleus of the cell. From the cytoplasm, to the nuclei. And you heard before that this is one of the most common mutations seen in patients with Waldenstrom. What is amazing about this mutation is a single change in one base alter the structure of the protein and make oncogenics. And it's one of the few cases in which about 90% of the patients do have this mutation. All other cancers that you may be aware, they do have other type of mutation, but they are not as common and as frequent as NYD88 in patients with Waldenstrom. So this is a unique situation, and it's a unique opportunity for people that we are interested in understanding the pathology and the biology of a protein to do this kind of studies. And although inhibition of NYD88 has been shown to be toxic to Waldenstrom cells, providing crucial insight for the potential of target therapy, these studies were limited, provide limited information as to the role of NYD88 in tumor initiation and lymphomagenic potential. Because this mutation was so highly recurrent in patients with Waldenstrom, people thought, oh, it's oncogenic. This mutation induced the tumor. And you will see that it's not entirely true based on our studies. So the first thing we tried to do is a mouse model of Waldenstrom carrying this mutation, the NYD88 mutation. And for this purpose, our hypothesis was that NYD88, the L265 mutation, is an oncogenic driver mutation. An animal model of LPL Waldenstrom can be generated by engineering a human NYD88 265 in mouse B cells. This is a challenging situation. It's not easy to do. I'm going to try to explain you here, because what we have to do is to alter the DNA, the genome of the mouse, so we can introduce those mutations that are seen in patients and animals. And the first thing that we did was to generate a construct, a piece of DNA that contains information so we can transfer into the mouse and into the mouse B cells. And we clone our protein. We cloned two proteins, the wild type and the mutant human protein in this vector. What we did then, we took this DNA and we transfected into, or we introduced inside the cell of mouse embryonic stem cells. And what we do after that, we identify that this mutation has been introduced in the genome of those cells, we inject those cells into blastocyst. The blastocyst is a little embryo that is going to originate the entire mouse later on. And then we take those blastocysts that carry the mutation and we inject in pseudo-pregnant females. And those pseudo-pregnant females, they're going to give rise to a progeny that contain this mutation. But the mutation is there, but it's not active yet. Why? Because we have engineered this vector containing this structure that is in front of the gene that we want to be expressed in the B cells. Why we are doing this? Because this inhibits the expression of this protein. Because we need to express this protein, we need to remove this, what we call a stopper. Once it's removed, we can have expression. Why we do this is because if you don't do this inhibitor, the protein is going to be expressed everywhere in the body of the mouse. And we want this protein to be expressed only in B cells. And a very particular type of B cell, that is the germinal center B cell, that is believed to be the cell that originates the tumor cell in patients. It's the germinal center B cell. Not every kind of B cell. It's one particular B cell. So that's the reason we are doing all this effort. And how we express this protein in germinal center B cell? We cross our mice carrying this mutation with a mouse that has been generated that is called AID-CRE. This animal has been engineered many years ago by other scientists. And what this animal is doing is expressing CRE is a recombinase. It's a protein that is produced in bacteria. It's not produced by human or mouse. But what is going to do that protein is going to recognize this sequence. And it's going to cut out this fragment of DNA, and we are going to have expression of the mutant protein NYD88 or wild type in germinal center B cells. So we did this a couple of years ago, and we can see here, we take a spleen from those animal transgenic mice, and we can see that the protein that I was referring to you, the CRE recombinase, is expressed in this nodule, in this cluster of cells. These are the germinal center B cells. These are the cells where Waldenstrom macroglobulinemia originate. And then what we did, using flow cytometry, we isolate those germinal center B cells using different markers. And you can see here we can isolate that, and it's a small population. In the spleen, in the lymph node, those populations are about 1 or 2% of the total cells in the spleen or in the lymph node. What we did then, we did PCR, is to identify, to see if this region that we wanted to delete in germinal center B cell is happening or not. And you can see here that in every cell, B cell that we isolated from both transgenic, the wild type or the mutant, we have a shorter fragment of DNA here compared with the wild type. So it's indicating that we are deleting this stuff and we are expression of the protein. In addition, we documented that this protein is expressed because we take RNA from those cells and we sequence. And we were able to identify the mutation. You can see here, this is the wild type protein and this is a change in base. You see there's a transition from thymidine to cytosine. So the system is working. After 2 years of work, we are able to get this first landmark in our research. So then we can use those animals to do biochemistry, something that we cannot do with patients. That's the reason this is important. All this work, we cannot do it with patients. So then we did Western blot. We prepared protein from a control animal or B cell that do have the wild type protein or the mutant protein. And we can see very striking difference. This is the wild type transgenic and this is the mutant. And you can see the level, the protein is much less in the mutant. And this is indicating that the protein is behaving differently. The mutant is behaving differently to the wild-type protein. In addition, what we observe, using immunofluorescence, you can see the nucleus empty. There are no P65 in the nucleus of these cells. But after transgenic, we can see that you can see those granules inside the nucleus. So our system is working. We have generated a mouse model, and it's activating NF-kappa B, that is the landmark of this mutation, and translocate a protein from the cytoplasm to the nuclei, and it's here. You see, it's not here, but it's here. Then, what else we identify? We did phosphorylation analysis. P65 is a protein that is phosphorylated, and we can see that the amount of protein phosphorylated in the mutant is lower than the other one. Another difference between the wild type and the mutant protein that we can identify in our system. What else we identify with this animal? You have here transgenic wild type and transgenic mutant. And we look at the protein expression of NYDA using antibody, and it's a technique that is called immunohistochemistry, and we look for the expression, and we can localize this protein in those cells. And you can see here that the wild-type protein is diffusely distributed in the cytoplasm, but the mutant is present in those granules, cytoplasmic granules. So again, another evidence that the mutant protein is behaving very different from the wild-type protein. Now what we are doing is trying to identify are the proteins that in some way are binding to the mutant, and so we can understand the biology of this protein. And as you can see here in a preliminary study in which we have all the protein that we can pull down using an antibody that recognizes NYD88 wild type, and we see all the proteins that are binding to NYD88 wild type, all this protein. But you can see also all the proteins that are binding to the mutant. And by comparing, for instance, this band, we see that here is not present and here is present. So NYD88 is also binding to different proteins in the cell. And this is going to help us to understand the biology of NYD88 and to develop new therapies. So going back to the characterization of our animal, young mice, we refer young mice about 4-week to 10-week-old mice. And what we observe is that this is the spleen and these are the lymph nodes. That in both transgenic, in the mutant transgenic, we see a slight enlargement of the spleen, but it's very consistent. So the tissue, the size of the organ is larger than the mutant and the non-transgenic. But we didn't detect any major histologic or immunophenotypic characteristic in those animals. There are subtle changes that we are still unable to characterize by using flow cytometry, immunohistochemistry. Probably the different are the level of the individual cells, okay? And one of the features also, at very early age, we didn't see bone marrow involvement with lymphoplasmacytic cell. That is the landmark of Waldenstrom. Something that it was very striking in mice, in these transgenic mice, is that wild-type animals look completely normal, but transgenic mice carrying the mutant protein, they do have alopecia in the neck. We have never seen this before. And then, so we characterize this by immunohistochemistry and immunohistological analysis, and we found out that there was an inflammatory process, very local in that particular, in no other part of the body there was this alteration, only in the neck of patient, of mice with this mutation. And it was characterized by a large accumulation of macrophages, and this characterized what is called Granulomatosis Dermatitis. Interestingly, last year somebody published a case of a patient, Waldenstrom patient, that has a mutation, somatic mutation in NYDATA, germline. And this patient had this rash in the neck that is characterized similarly to what I've described, what you see in the mouse. So again, this is indicating us that our mouse animal model is behaving in the situation we expect that will happen in human. The problem is that for some reason, this alteration is not highly recurrent in patients with Waldenstrom, but is highly recurrent in mice. And you may understand that these are different species and the immune system behave in a different way. So those animals that do have a skin alteration, we did flow cytometric analysis, and what we observed in the spleen and lymph node of this animal, young animal, there was increase in the number of germinal center B cell and also in plasma cells. And in addition, what we observed that there was an increase in IgM. We have been hearing before that IgM spike is very characteristic of Waldenstrom macroglobulinemia. So our animal also overexpress IgM. And you can see here that this animal also do have a large increase in the level of interleukin-6 in the serum. That this is another characteristic of patient with Waldenstrom. Probably you haven't heard about that because this is more at the experimental level. So then what we did, we took a cohort of a mutant transgenic animal and wild type, and we compare the survival of those animals over a long period of time, up to almost 2 years. This is old mice equivalent to the old human being. And what we observe that only those animals that do have the mutant protein became sick, and they were characterized by a lymphoproliferative disorder, enlargement of spleen and lymph node, and accumulation of this very unique type of cell that I'm highlighting here, that these are lymphoplasmacytoid cells that do express markers for B cells and for plasma cells, CD138. Because this is a cell that has a behavior that is in between a small lymphocyte and a large cell that is the plasma cell. So we have those lymphoplasmacytic cells in our animal. The only issue that we don't have is that those expanded cells are not clonal, and in patients with Waldenstrom are clonal. What is telling this? This our model recapitulates several of the clinical features of Waldenstrom, but we need something else to make this cell tumorogenic, to become very malignant. And one of the landmarks of malignancy in hematopoietic or lymphoneoplasm is clonality. Clonality in the germline of the immunoglobulin gene. This LPL was also characterized by flow cytometry, and we observed that there were increase again in plasma cells and in germinal center B cell. And most remarkable is that those animals with the mutant but not the wild type transgenic develop what we could say a spike, but it's not a spike, it's not a narrow band of immunoglobulin, it's a broad, indicated that it's a polyclonal process. We don't have only one cell that is expanding or taking over the tissue. There are several cells that are becoming increasing numbers. Then we observe again IgM is increased, and again we observe that the mutant mice develop what I mentioned at the beginning, the rouleau formation. And this is related to the hyperviscosity syndrome. When you have too much IgM in the blood, the red blood cells, they do not move freely and they get stuck, and then you see this aggregation of cells. So our mice are also developing this very peculiar characteristic of patients with Waldenstrom macroglobulinemia. I'm going to mention another very important analogies between our mouse model and the human. I mentioned before that interleukin-6 is increased. The level of serum interleukin are increased in patients with Waldenstrom as compared with normal patient. The same happened in our mouse. We see increase in interleukin-6. And something that also been described in human is that CXCL13 is increased in patients with Waldenstrom, but compared with normal. And we see the same in our mice. Then, if you remember, I mentioned that the mutant protein aggregates in the cytoplasm of our transgenic mice. So then I went and see what happened with human. So I took patient samples, and I did section, and I stained with antibodies again, NYD88. And look at this. I can see that the mutant protein produced those little aggregates, but not the wild type. Again, another similarity between our mouse model and patients with Waldenstrom. This aggregate had been identified before for somebody working not in Waldenstrom, but working in diffuse large B-cell lymphoma that also has this mutation, the NYD88, but it's less frequent. It's only 40 or 30%. And he found that NYD88 in those patients with diffuse large B-cell lymphoma aggregate in the endolysosome. We believe that in Waldenstrom, NYD88 also localized in the endolysosomes. And this is something that's not been described yet for patients for Waldenstrom. This is unpublished work. Something else that we identify in our animals is that there was an increase in the mutant of T cells. Somebody was talking about how important is immunology in Waldenstrom macroglobulinemia. This is an evidence that immunology is important. We see an increase in T cells in the bone marrow of our mutant mice, but not in the wild type. And I went and I looked at TMA, a lot of samples from patients, and I could see again that T cells are also increased in patients with Waldenstrom, similar to our animal. Another important feature, our animal, we see an increase in mast cells. Somebody was asking me today, what has to do those mast cells in Waldenstrom patient? They are increased, but probably they are not oncogenic, they are not tumor cells. These are bystander cells, because the B cell secrete chemokine like interleukin-6, like CXCR13, and attract the mast cell to the bone marrow and to the tissues. This is the reason we see increase in mast cells in our mutant transgenic NYD88 mouse and in patients with Waldenstrom. We also observe in less than 10% of the animals with the mutant protein, not with the wild type, that some of the animals develop a very aggressive disease. And as you can see here, this is the spleen of one of those 4 mice that in our entire cohort. It has a huge spleen. And immediately this is telling you something very wrong is going with this animal. And there was also enlargement of the lymph node. And by histopathology, what we observed, that there were not the classical LPL cells, the lymphoplasmacytic cells, but there were these very large cells, ugly cells, terrible cells. And they were B cells, and they have a high proliferation index. The cells, these were growing like crazy. And this was consistent with a diffuse large B-cell lymphoma. What is telling us? That our animal model developed this premalignant condition, this resembling Waldenstrom, but progresses to diffuse large B-cell lymphoma, like in patients, unfortunately, some of the patients evolving to diffuse large B-cell lymphoma is happening also in our animal model. Again, I want to tell you that I believe we are in hand of a very good model or pre-malignant model of Waldenstrom macroglobulinemia. And those larger cells, as you can see here by flow, they are large. And the most interesting, I was talking about clonality. That before early age, this animal developed a lymphoproliferative, it was non-clonal. And you can see here, these large cell lymphoma are clonal because you see additional bands in the B cells. So we did a whole genomic sequencing from those animals that they do have this large spleen with large cells, and we identify mutation in genes that have been seen in human patients that do have de novo diffuse large B-cell lymphoma, or a patient with Waldenstrom that progresses to diffuse large B-cell lymphoma. Again, our mouse model is resembling a lot to the human condition. The only issue that we have is that the disease is not clonal. So my conclusion for this part of the talk is that NYD88, the mutant one, is insufficient by itself to promote neoplastic transformation in mature mouse model. And this was not known before this work. Everybody thought, oh, this is oncogenic. This mutation is going to produce tumor. Helps to produce the tumor, but does not produce the tumors. The NYD88 L265 mutation promotes a non-clonal B-cell lymphoproliferative disorder with many features of LPL Waldenstrom. This polyclonal disorder can transform into aggressive large B-cell lymphoma at low frequency and long latency. And transformation to diffuse large B-cell lymphoma is associated with acquisition of secondary genetic lesion frequently seen in de novo human diffuse large B-cell lymphoma, as well as lymphoplasmacytic lymphoma can transform to diffuse large B-cell lymphoma. So we believe that due to the development of a pre-malignant condition and long latency and high penetrance, because most of the animals develop this problem, this lymphoproliferative disorder, we believe that the NYD88 mutant transgene constitute an ideal base model with which to build up a more refined mouse model of LPL Waldenstrom. What we are doing now? So our hypothesis is that we can engineer a refined mouse model of Waldenstrom by co-modeling in the mouse two of the most common and functionally associated genetic lesions seen in patients with Waldenstrom. NYD88 plus chromosome 6 deletion. I think nobody was talking today about chromosome 6 deletion, but we have data from Dr. Trion's lab and other investigators that chromosome 6 is deleted in about 50% of patients with Waldenstrom. And other patients do have partial deletion of that chromosome 6. So about 80% of patients with Waldenstrom do have in some way deletion of genes that are localizing in chromosome 6. What is interesting is that in mouse, we got lucky. All the genes that are located in chromosome 6 are located in chromosome 10 in mouse, what is called synteny. So chromosome 10 in mouse contain all the genes that are deleted in patients that do have chromosome 6 deletion. So now what we are doing, we want to generate a mouse that has a big deletion of chromosome 10 that is equivalent to the human 6. And we have been successful on that. This is a very, I would say, difficult project because it's a long region of chromosome 10. 30 kilobases. Nobody, I'm not aware of anyone in research has been able to accomplish a deletion as large as this one to delete a gene, a large chromosome region in animals. We have tested this using PCR technique that we use in the lab to detect this deletion. The deletion is present in embryo, in mouse embryonic stem cell, But then we cross those animals with a decrease, and we also see a decrease of the genes that are residing in chromosome 10. So we have in hand now again a mouse model of chromosome 10 deletion. And next step, what is going to be to cross our NYD88, a mutant protein animal with chromosomes 10 deleted, and see if they develop Waldenstrom macroglobulinemia. I think now I'm gonna thank everyone that has been made possible this work, but in particular to the Waldenstrom Macroglobulinemia Foundation of Canada that has given us generous support for making this work possible. I hope you understand. I think it's for a clinician, it's very difficult to understand sometimes what is the meaning of having people working in the lab trying to generate this mass model. I have to tell you that I do believe, and please believe me, that this work is important. Thank you so much for your attention. If you have any question.
Does that have any backing into, we were talking earlier about the DNA? Can that backtrack into them linking up and looking at a DNA?
Oh, yeah, you are. I think you are trying to say if we can edit the NYD mutation. I think now there's a technology that is called a CRISPR-Cas9. What people are trying to do is to reverse that mutation, from the mutant to reverse the protein to wild type. It's a technology that is available and is in place. And I'm telling you, in 10, 20, or 50 years from now, the technology is going to be possible. We are going to be able to re-edit the genome of each one of you and reverse the pathology. This is possible. This is molecular biology. This is genetic. This is possible. It's going to be possible. Did you see the last picture of the black hole?
Yeah, I saw the picture of that.
It's incredible. Nobody has been able to prove that.
Yeah.
Nobody's been able to prove, but it's going to be possible. I think we are going to find a cure for cancer.
I think there's a little test that we're just going to pass out. It really is pretty thrilling for us at the WMFC to be funding such fundamental research and to see it coming live and is going to make a difference, not just for Canadian WMers, but for WMers all over the world. So thank you very much, Ruben. It's great to have you here. We appreciate all your work.
Thank you so much. Thank you to all of you.